pleasure to introduce. Yes, I'm not sure what we should do about this. How about now? No, they can still hear me.
We can hear you fine and we can see the presentation fine.
Okay, can you hear the echo though?
No, for me it's okay.
For us there is an echo. Okay. Yes, it's a great pleasure to introduce Dr. Kyriaki Michailidou, who is a statistical genetic epidemiologist with extensive expertise in the analysis of large genetic and genomic datasets. She is currently an associate scientist and lead of the biostatistic unit at the Cypriot Institute of Neurology and Genetics. She's trained in mathematics at the Aristotle University of Thessaloniki, applied statistics. He did her master's at the University of Oxford, and genetic epidemiology at the University of Cambridge, where he did her doctoral studies. Her research lies in the fields of statistical genetics, epidemiology, and genetic epidemiology of diseases, with a focus on the analysis of common and rare genetic variants identified through genome-wide association studies and next-generation sequencing experiments. She has been involved and led the analysis of some of the greatest GWAS in breast cancer conducted by the Breast Cancer Association Consortium, and identified more than 100 of the current known polymorphisms that are associated with breast cancer susceptibility. She has authored more than 150 publications in high-impact journals and is the leading author of publications in Nature, Nature Genetics, Cancer Discovery, American Journal of Human Genetics, and others. I'll leave. Yes, okay. Okay, yes. All right, I'll leave the stage to you, and thank you for coming today.
Kyriaki, we can't hear you. There we go.
We need this to be green.
Okay, can you hear me now?
Yes, we can.
Okay, we got rid of the echo, which is good. So, hi everyone, it's a pleasure to be here. I'm Kyriaki Michailidou at the Biostatistics Unit of the Cyprus Institute of Neurology and Genetics. And today I will talk about Genome-wide Association studies and breast cancer risk prediction. It will be a bit of superficial talk on them, just to give you an overview of what we've been doing. And if at some point anyone wants to stop me, I'm more than happy to stop and explain something more, because I know we are from a wide variety of backgrounds. So I'll try and do my best to explain what we're doing. So breast cancer is one of the most common cancers in the world. It's the commonest cancer in women, and is determined by important lifestyle and genetic determinants. So these different important factors altogether contribute to the architecture of the disease. They're both germline and somatic variants that have been identified as associated with the disease, also common and rare variants. In my talk, I will focus mainly on the genetic determinants of breast cancer, on germline. Basically, germline is what you carry with you when you were born, whereas somatic is something that you get through your life. And I will focus on common variants, and I will come back to that in a bit, what do I mean by common variants. And I will touch a bit upon how you can use this these sort of genetic factors in order to inform screening and prevention strategies in general. So just to give you a bit of an introduction and historical introduction of the genetics of breast cancer. So here you can see a pedigree. So quite early on, it was observed that some families they have a lot of breast cancer cases. So some breast cancers run in families. So this, just for those that are not familiar, so this is a pedigree. It's like a family tree, basically. With a square dot, you can see a male. With a circle, a female. And this is they're connected, and the line, you can see the offspring. So these are the children of these two individuals. And then with a crossed line on top of those circles and squares, is an individual that has died. And then with shaded black, you can see different cancers in the family. So the majority of those, if you see they're breast cancer. So you can see these two individuals have two daughters. They have 5 daughters, and all of them have breast cancer. 2 of them at a really early age, and they had 2 breast cancers at the age of 33 years old. So at both breasts, basically. So this was one of the observations that led to the hypothesis that cancer runs in families. And in order for that to explore what happened, they thought that there must be something genetic, something that runs in those families and is actually causing this excess cancers. And just to show you, sorry. What are the numbers there? The ages. So it. This one 3, 4, 4. This one are just the numbers of the family members. It's just so this is the first generation, and then the second generation, then the third generation, the fourth, and the fifth. No, it doesn't. I mean, usually it's who is actually the first individual that has been tested, and then all the rest of the family members. These numbers. This is just from the original publication in 1994, which was actually the publication that they localized the BRCA2 gene. So for breast cancer, there's 2 really high risk genes. So individuals that carry variants in those genes, they're at really excessive risk of developing the disease in their lifetime. So this is how they identified originally those 2 genes. So I don't know if you've heard before, like the Angelina Jolie story, that it was identified that she had a genetic mutation, that it was actually increasing her risk of breast and ovarian cancer. That's why she went on to perform prophylactic surgery in order to reduce her risk of developing the disease. So this is just historically how it all started from this observation. So this is the current landscape of breast cancer genetics by the end of last year. So these are the really high risk genes that I've showed. So they were identified, BRCA1 and BRCA2 were identified back in the 1990s. And there's also some other high risk genes. Some are part of different multiple cancer syndromes. So it's not just breast cancer, but there are families that they have more than another different sort of cancers. And then basically what it was shown over the last decade and the last 15 years is that although there's these genes that are actually causing a really high risk to the individuals that carry them, the majority of cancers, of breast cancers, as you can see here, are actually unexplained. They don't carry these variants in BRCA1 and BRCA2 and the other genes. So this is actually what caused the shift over the last 15 years from these sort of studies that are pedigree studies, family studies, which I showed you earlier, and these were used to identify these rare variants in the population that caused a really excessive risk in the carriers. They moved to a shift called now association studies, which are actually studies performed in cases from the population that are not actually related between them. It's not in families. It's basically random cases in the population, and you try to explore what might be different between the two set of populations. So, sorry.
Can I ask, when you say high risk, how high is that risk?
It's like 10 times more usually. I mean, on average, it's around 10 times more likely to develop cancer compared to the general population. So if breast cancer is 1 in sort of 10 or 8 in the general population, yes, over their lifetime, then that's I mean, by the age of 80, some of those might have even like a 60 or 70% chance of getting breast cancer. So it's quite high. And then, so this is just to show you, so you see that these are really rare and they contribute a risk compared to the general population up to 10-fold. And nowadays, genome-wide association studies, which I will focus in a bit, we are in this end of the spectrum. So this is what we're going to talk about. So these variants are common in the population. They're found in everyone. Each individual one does not contribute an increased risk of as high, but then if you put them together, you have the potential of being used and have used as a risk prediction tool. So, but just to go a step back in order to explain what do we mean by this genetic variation and how that works. So if you have two different individuals and you scan their DNA, so I presume you've heard a lot of the times that we have around 3 billion base pairs on our DNA code. So you can genotype, so getting a blood sample or a saliva test, and then you can genotype two different individuals. And you will get this the DNA. So the majority of these 3 billion base pairs are the same, but then we have a lot of locations that they're different. So a single base pair difference at a specific location of the DNA, that's a SNP. And that's our unit of interest, and that's what we explore by these studies. So and you can consider, I guess, a lot of different of you are exploring different factors in terms of diseases or in terms of other determinants. So is if you're exploring if pollution is associated with certain diseases. So here, and you see that individuals that live at polluted areas have more high incidence of the disease compared to other individuals. So it's the same situation, but you compare the frequency of this DNA change in the individuals that have cancer compared to the others that don't.
But this is purely genetic, so it doesn't get done far.
No, so these studies are mainly focused on genetics. So, and then, yeah.
So SNPs, would you find them in all cell every sample?
So this is, yeah, that's why I said that this is germline. So this is what you find in all your cells.
For example, the somatic ones, if you take the samples from different positions in the body,
yeah,
you have different.
Yeah, usually somatic you refer usually to tumor samples. So if you get a sample and the DNA of a tumor, it will be different than the rest of the cells of the body. So, but this is mainly from blood.
So this is for all the cells in the body have this thing.
Yes, yeah. We assume that that's true. I guess you could debate, but I guess that's the hypothesis. And then, so this SNP, so as I mentioned, so we have this 3 billion paper, 99% of our code is the same between two different individuals, but we try to find that small percentage that differs. But by small percentage, I mean millions and millions of these variants. So genome-wide association studies, which is the way that we actually use in order to explore these variants, evaluate hundreds of thousands to million polymorphisms. And by SNPs, we mean SNPs that are common in the population. So you find them in more than 1% of the population. And then usually how these are conducted is by getting DNA samples from healthy individuals, which are called controls, and individuals with a disease of interest. So it could be, you might be interested in blood pressure or height or BMI, or you might be interested in a disease. So depending on what's your phenotype of interest, then you will do that exact analysis. And this can be done by genotyping using various SNP arrays. Then you compare the allele frequencies between cases and controls. And again, this is what I explained earlier. So it's as if you are exploring some other risk factors like smoking or pollution or so forth. It's just because we have so many of those, we need to take the specific features of this genetic data into account when we're doing this analysis. So these are actually possible to conduct. Sorry. These are actually possible to conduct because technology has evolved and there's some commercial arrays that are actually can be used. And I mean, you can actually sort of do it with 50 euros per sample. Whereas if you consider the original sequencing and so forth, it took maybe some billions in order to do one individual for the whole genome. So these are quite cheap nowadays, and that's why you can actually do a lot of different samples. So sometimes these studies are conducted in 2 stages. So the first stage, you explore all the SNPs, millions of SNPs, and then you get the ones that are significant, and then you try and replicate them into different studies. So this is just to illustrate that GWAS have been performed in a lot of different diseases and traits. It's not a great resolution here, but so these are the chromosomes. So here you have the chromosomes, and with circles are associations that have been identified in different diseases or traits. So there are around 350 variant trait associations on the GWAS catalog. So you can go on the GWAS catalog, is open and free. If you're interested in a specific location or a specific disease, you can actually see which variants have been identified and are associated with a specific phenotype.
So this association is driven statistically.
Yes.
So based on some specific criteria.
Yes. So yes, I think this is my next slide, maybe in a bit. Yeah. So basically, you have to have quite stringent. So because you're doing so many different tests, you have to redefine your statistical significance. It's not because a usual p-value is 0.05, for example. You cannot really use that because you're going to have a lot of false positives. So it depends on the tests.
What are the chromosomes?
Because there's so many associations. So sorry. So you can see here with lines. So this here is each chromosome, and then you have, maybe I should have made it a bit better, but you can see here, these are the chromosomes, and this is the location where each variant has been identified. I can't.
On the left and the chromosome.
Yeah, this line, I mean, it's not a great picture, but you can see here, this here is a chromosome, and then you have out just where because you have so many different associations, you cannot really see them. Yeah, go ahead.
How do you know which one is kind of the standard chromosome and then the
What do
others
you
are
mean?
different?
That's you comparing between the control. So you assume that so it's on average. So you have lots of different individuals. So you might have thousands of individuals in your analysis. So you compare between the healthy individuals and the individuals with the disease, and that's how you find the difference in association. But yeah. So, but because in order for these studies to be feasible, you need really large sample sizes. So it means that you need a lot of individuals to be part of these studies. And this is where Breast Cancer Association Consortium comes in place. So this was formed in 2005, a lot before I started. But so the Breast Cancer Association is a consortium of researchers, clinicians, and a lot of individuals that are interested in breast cancer research. Is the biggest actually cancer consortium for association studies in the world. And it's formed by different groups in a lot of different countries. And the Breast Cancer Association Consortium has conducted some of the biggest genome-wide association studies in breast cancer. One project was called iCOGS and the other Oncoarray. And in total, more than 100,000 individuals cases and 100,000 controls. So these are from the different countries and from different ethnicities and so forth. So this is just the progress of breast cancer GWAS, and this is also mainly on the cost. So the first GWAS was published in 2007, and that was on 400 samples, so 400 cases and 400 controls. And over the year, due to the cost and due to these collaborations that were formed between the different groups, they were able to perform large-scale GWAS, like the iCOGS and the Oncoarray, which was on more than 120,000 samples. And that currently we are part of this study called Confluence, which is an NIH-funded study, and aims to actually genotype 300,000 women and some men from all over the world, also to increase the diversity in these genetic studies. So what do we know about these common risk alleles? Is they're mainly identified through genome-wide association studies. More than 200 of those were identified by the end of 2021. The majority of those were part of the Breast Cancer Association Consortium. This studies the COGS and the Oncoarray. And this is just a sum of the previous slide, but we are aiming, we are currently part of this study that we are hoping that it will identify even more variants that will be used to inform risk prediction in the future.
Sorry,
Yeah.
how does the risk evolve throughout the years? Or,
You mean?
how many? Yeah.
Okay, sorry.
Susceptible for.
So basically, I mean, in the first round, originally they identified few of these variants. So like the first publications were like 5 variants. Then the iCOGS study identified 41 variants, and then the Oncoarray project identified 65 regions that are actually associated with breast cancer. So this comes back to the question of the analysis. So usually, you compare the frequencies of the SNPs between your cases and your control, so the healthy individuals and the individuals with the disease of interest. Usually we use logistic regression, and you adjust by study, principal components, by age, and other sort of different variables that could be confounders. And then you get a p-value. So usually you get that for a single SNP. So you might have millions of these SNPs, and you test them each individually in respect to the disease, and you compare the frequency between cases and diseases. And then this is where the p-value, you really have to have a really small p-value. So this is practically 0 if you consider how small this value is. But this comes in because we need to have a really stringent threshold in order to reduce the false positives. Otherwise, if you get the 0.05 and you do 1 million of tests, you will get 50,000 associations just by chance. So you need to have this really strict, very small p-value.
For the frequencies.
Yeah, because you are actually exploring so many different SNPs, and you explore them. So this is just to show you how do you visualize the results from the analysis. So if we just go to the plot on the left, so this is called the Manhattan plot because of the the peaks is sort of like a skyline of Manhattan. So this is actually you're plotting the -log10 p-value. So for each individual SNP, so each dot represents a SNP, sorry, and on the x-axis is the chromosomal location of the different SNP. And by the peaks here are the variants that are significantly associated with the disease of interest. So this plot actually has 11 million dots. So these are 11 million variants that we assessed and we explored. We did the analysis and then we plotted them. And you can see here that this is a SNP. I mean, the top one is the most significant one, but it's sort of a region that is associated. It's not a single individual SNP. And that adds to the complexity of GWAS, because when you want to move from statistical association to identify what is actually causing and how is actually the functional sort of mechanism behind the disease, you have to actually go into more detail into the different genetic regions on the different chromosomes. So, for example, I'll just illustrate one region, which is on chromosome. Yes?
What's the difference between the clone and female?
Here, you mean? It's just to separate the different chromosomes.
Okay.
So because it's just, sorry, it's just as a visualization because it's too complex. Otherwise, it would have been impossible to distinguish.
Chromosome has how many positions?
That's quite variable as well. So the biggest chromosome is chromosome 1 and 2, basically. So you have a lot, and the smallest is chromosome 22. So for example, I can't recall exactly, but I think, like for example, chromosome 1 might have 1 million SNPs.
Okay.
So I think
It would be 1 million base pairs.
yes, not just 1 million base pairs. It's 1 million base pairs where you have differences between which are different between two pairs of individuals, because you will have a lot of base pairs that are completely the same between everyone. So you won't get any information if you get those.
So how many base pairs do you have in the chromosome? On the SNPs?
I'm not 100% sure on that. Yeah, I mean, the total code is 3 billions. So I'm not sure exactly how they divide into the different chromosomes. But I mean, the chromosome 1 and 2 are the biggest ones, and chromosome 22 is the smallest. And the X and Y chromosomes are small as well. So this is just the location. I mean, the location, and then the more significant the variant, the biggest height of the peak. So, and also to add to the complexity, breast cancer also have some subtypes. So you have estrogen receptor negative and positive breast cancer. So I'm not going to go into much detail into that, but this is just to illustrate the differences in the genetic etiology. Even within breast cancers, you have different separate diseases. So, and then this is, so if you go, for example, to one specific region, for example, here, so you see this peak, but if you zoom in the region, you will get this thing. So this is one, this is just one region that you find a significant SNP. And then if you zoom in, so if you sort of zoom in, then you get this region, and you have these all these SNPs that, so you will have different locations that are associated. Here underneath, you have the genes. So you don't really know, so you have the statistical association, and when you want to go forward and see what is going on and how this is causing development of the disease, you have to actually link these variants to a specific gene and to the function of the gene, and see how this actually is going into causing breast cancer. So what we tried, so this is another complex part of the data that we're trying to do, is trying to actually predict, so moving from the statistical association to the functional mechanism that is actually causing the disease etiology. So one of our colleagues, Jonathan Beasley at the QIMR, developed this pipeline called Inquisit, which is actually you try to predict using not just the genotype information, but other data from public databases like the MetaBrick, the TCGA, and other functional data sets that are available, in order to see how these SNPs regulate gene expression in order to start carcinogenesis, for example. So, and what we found from all the different studies is quite a lot of these SNPs are regulatory. So they might not be in the gene, they might be away from the gene, but the way they might be actually working in order to explore, to differentiate the gene expression and then cause breast cancer. So what have we learned with the GWAS over the last 15 years is that, so this was a plot that we had originally with BRCA1, BRCA2, and the other high-risk genes. These are the ones that we can identify with the current studies. And what we've actually also shown is that having a bigger sample size and larger studies, you can lead to being able to identify another 18% of this familial risk. But still, you can see that we have another 40% that is still unexplained.
How do you know that this will be 80%?
Sorry, this was actually based on estimations from the current data, and saying so if you increase your sample size, you can see how many SNP. I mean, it's an estimation. It's an extrapolation based on the current data. So we're hoping with the Confluence project that we are going to be able to identify more variants and explain more of this familial relative risk of cancer, of breast cancer. Sorry.
So could be unexplained ones due to somatic or other reasons. I mean, is it saying that is unexplained, cannot be explained by germline?
I mean, there could be a lot of explanation. It could be that there's some rare variants that have a small effect similar to the SNPs that we won't be able to identify unless we have millions of samples. It could be some due to like environmental factors that we know that exist. So it could be shared environmental factors within families. So there's lots of hypotheses, like trying to see what could explain those. So what's the time? Okay, so so that part was the first part, and the identification of the variants and how we can actually use these studies in order to identify these genetic variants. And then, so one current research and is quite fashionable, I would say now, is something called the polygenic risk score. So basically what we're trying to do is to see if we can put together all these variants, because I mentioned they are all contribute a really small risk individually. So if, for example, I have one of the risk variants, even one of the most strongly associated ones, is the odds ratio is 1.3, meaning that you only have like a really small increase in your risk. So individually, it doesn't say much about your risk of the disease, whereas where you have one of those variants or not. So what has been developed is something called the polygenic risk score. And basically for each individual, for each woman in this case, because we're talking about breast cancer, you actually sum up the number of risk alleles an individual carry for the specific variants that we identified. So for example, here we then is the total number of SNPs. And then, so this is the estimate from the logistic regression. When you do logistic regression, you get the effect size. And then it's the genotypes for each individual. So for each genotype, you might have 0 risk alleles, you might have 1 risk allele, or you might have 2 risk alleles. So this is sort of a sum of your risk. And then, so this is just also an illustration. So if you assume these are the chromosomes, here like the 22 chromosomes and the XY chromosome, one individual might have only 1 of these risk allele in their genotypes, in the germline, whereas another individual might have all of those. So basically, when you scan your DNA using blood or saliva, you can actually calculate this risk. It's similar. I mean, what we're trying to do, I mean, what like a lot of studies are trying to do is get this score, and it would be similar to if you get your blood pressure or your estimates about your, I don't know, cholesterol. And this will give you a risk and estimation of how likely is to develop coronary artery disease, for example. So similarly, but more complexly, you can actually get this score, and it will give you an indication of your risk of developing the disease. So this is what it was observed, that if you get this polygenic risk score, so this, I don't know if you're familiar, but probably you are. So it's like a normal distribution, basically. So the majority of the individuals will have an average risk and will be in the middle. So we will not have a lot of the risk alleles. Some individuals will have none of the risk alleles, and then a small fraction of the individuals at the upper tail of the distribution will have high risk of developing the disease. So what different studies are trying to do now is see if we can identify these women, will it be more cost effective and more beneficial in order to target those women in terms of risk prediction. And then what studies others have shown is that you can see if you get the distribution of the risk alleles in women that have breast cancer and women that are controls, you can see there's a shift in the two distribution. It doesn't seem big here, but if you go into the most like the numbers, because there's so many women, is actually a big proportion. So you can identify those women that are at the tails and upper extremes, and that will lead to a better discrimination between cases and controls. So this is another study by the Breast Cancer Association Consortium that we actually tried to develop a polygenic risk score and see what was the best way of identifying these variants. So we did the GWAS, you get using the p-value. But then we thought of exploring other methods like lasso. I don't know if you've came across. So it's like a shrinkage method. So rather than getting the p-value alone and get only ones that are significant, lasso actually uses different methods of getting this shrinkage. So actually how to identify which variants. But actually, like using the hard threshold thing, which was just based on p-values, you can see that the model was slightly better with the lasso, but you have a huge increase. It's like 10-fold increase in the number of variants that you need to use in order to get that that difference. And in the AUC, which is the model calibration, it wasn't extremely much better. So we decided to go with the 313 PRS. So with 313, it means that we're using 313 regions, SNPs on the genome, and then you actually calculate the PRS polygenic risk score based on those variants. And so this again is from the same publication, illustrating what's the potential benefit of this sort of screening. And so this is the 10-year absolute risk of developing the disease. And these are the age distributions. And here is the percentile of the PRS distribution. So getting the individuals that are at the extreme of the distribution, which we showed like the normal distribution. So you get the individuals that are the extreme, and then the different cutoffs. By red line here, so this red line here is the average woman's risk at the age of 50, which is currently recommended to do to start having a mammography. So you can see that although this is the average risk, some women reach that risk at the age of like a bit after 30. Whereas some other women never reach that risk. So it will be more cost effective and more cost efficient for women as well, not just in terms of money. It will be better and a better strategy rather than just having at 50 years of age, everyone having a mammography in order to find a way that you can actually use this information to inform screening strategies. And again, what it was shown by different studies is that the PRS, the 313 PRS, can actually be used to discriminate between ER-positive and ER-negative breast cancer, which are two subtypes of breast cancer. It can also be used to predict the risk of developing a second breast cancer after you have a first breast cancer. It can actually also be used to discriminate between, like, so you will get a risk of women that are more likely, if you have a high PRS, is most likely to get a second breast cancer in your life. And again, what it was shown as well is that although carriers of mutations in BRCA1 or BRCA2 are at high lifetime risk of developing the disease, when you actually discriminate, so if you use the information of the pathogenic variants in these high-risk genes, but together with the common variants, you can actually see again this sort of discrimination in risk. So you can actually see, even with the individuals that carry pathogenic variants in those genes, you can actually distinguish their risk based as well on the SNPs. So what was else shown? So, and maybe I should have mentioned it earlier, but the majority of the studies that were conducted up to now and what I've shown were based on women of European ancestry, which is one of the big criticisms of the GWAS and so forth, that they've been conducted, all the genetic studies on women of European ancestry, and there's a big trend now trying to close this gap and see if this identification of these variants actually works also on other ancestry individuals. So it was shown that the PRS actually works for women of Asian ancestry. It doesn't work as well as in women of European ancestry, but it actually also predicts breast cancer. And what was shown from the different studies in Asian ancestry is that even within Asian studies, if you have different subpopulations, this is actually different. So moving from that, we wanted to see, so and I will touch upon some studies that we're currently working on at the biostatistics unit. So we've seen that in women of different ancestry, in women of different subpopulation in Asia, the polygenic risk score is different across the different ethnicities. And we wanted to see if actually this happened. I mean, if you classify everyone as European, we wanted to see if actually that has also differences within Europe. So, for example, this question actually started. So, for example, I was living in the UK. If I go to a UK in a clinic that are actually implementing risk stratified clinic screening for breast cancer, would my risk be different compared to the average UK woman. So I wanted to see how that actually changes. And this is work at the biostatistics unit by PhD student Christy Ayangu. So what we tried to see is using data from the Breast Cancer Association Consortium, or around 100,000 cases and 100,000 controls. We wanted to see how this risk actually changes within countries in Europe. So rather than grouping everyone as European or European ancestry, seeing how that differs. So this is something that is actually quite fascinating, and it's been shown by other studies as well. But so here you can see a principal component map. I don't know if you probably came across principal components previously. So it's a dimension reduction from the SNP data. So you have the SNP data, which are like 40 or 50 or a million variants, and you try and plot it in 2D, and it's a distance between the genetic distance, so between the different individuals. So here we're just plotting, the only information is the SNPs and the country that the individuals come from, the women come from.
You've only put here.
So yeah, it's only the two principal components, the first two principal components. So you
Just curious, how much different is the background?
I mean, so basically the first two are sort of the difference in the European sort of north and south. So you can see here, so here are the individuals plotted, and you can see that it sort of reflects the map of Europe if you think about it a bit. So you have here, sorry. Yeah, so it's Israel, Greece, and then you go up to Finland, Denmark, and so you can see that it actually, even just by plotting the genetic SNPs, so nothing else, just the frequencies of the SNPs, so you see that it actually you can actually cluster the different countries together. So this we thought maybe this will have some implications into actually the risk prediction when you get to the point of using overall estimates. And this is actually true. So basically, we try to see the polygenic risk score in the different countries. And you can see, I mean, long story short is that quite a lot of the countries, they cluster around the overall mean. But you can see that women from Greece and Italy, they're a bit shifted to the right of the distribution, whereas women from Ireland are on the other side of the distribution. So what we're trying now is ongoing work. So what we're trying to do is to see how this can actually be used to inform the screening strategies. So this is, what does it mean basically, is that women of Greek origin, they're sort of going to be a bit misclassified. So if I go to the UK clinic and they use the UK average, there might be a bit of misclassification at the edges of the distribution. And the same for Italy, a smaller misclassification, and for Sweden or Ireland is the other way around, that you have a small underestimation of risk. So now we're trying to see how we can actually use this to inform the general strategy. And the last part is also work from Christia, is that we actually use the PRS to see how it works in the Cypriot population. So we have a study called MASTOS at the Institute on around 1,000 cases and 1,000 controls. And we only had information of 15 SNPs because it was an older study. But we actually wanted to see how this can be used, and if actually putting all these SNPs together has some predictive performance in women of Cypriot origin as well. And this was published recently. What we also did is putting together the classical risk factors and the genetics in an integrative model. And what we showed here is that actually it works. So you can see the distribution is not as wide because we don't have all the SNPs, but it is actually on the right direction. So when we are going to be able to see and have all the SNPs, it would actually, we hope that it will work better. And this is basically the sum of what these studies are trying to do, is that what you want to put together the classical risk factors for breast cancer, which are sort of parity, age, other risk factors such as alcohol consumption, age of first period, age of menopause, and so forth, and put those together with their genetic factors and see if that's a better strategy compared to the current strategy. And some theoretical studies have shown, this one. So some studies, also part of the Breast Cancer Association Consortium, but and other collaborators, they have shown that if you actually use an integrative model that uses all the risk factors plus the genetic factors, you can actually and get the women that are at the highest 3% of the distribution. For example, a study in the US has showed that actually contributes to 3.5 million women, but you will get the larger percentage of breast cancer in that sort of subpopulation. So by having a more intensive screening in that set of individuals, then you will have a better model. So you want to move from just having age as the baseline for starting your screening for breast cancer, put this individualized component based on the classical risk factors and the genetic factors, and move to different recommendations for screening. And the current studies, like big studies in the EU and the US, that are actually trying to see if implementing such a strategy is actually works well. So. And we're currently actually working on some fine mapping, some rare variant association methods, and we're using Castor-C resources for some of the computational part, and Meluxina access. We also have access to Meluxina that the Euro HPC, and we thank the Cypriot Institute for their help. And just to thank the Breast Cancer Association Consortium, which provided the data, and this was from a meeting in 2017 in Limassol, and this is the biostatistic unit at the Cypriot Institute of Neurology and Genetics. And just to say that some of these slides are from Doug Easton, so to thank that as well. Thank you.
Okay, thank you. This was a fascinating talk. Let's start with questions. Any questions from the audience? Actually, you can.
Okay. Yeah. So, are there any questions? Was everything very clear?
We asked many things. So
No, it's fine.
Just from curiosity, let's say two questions. So, so the first is from the modeling point of view. You share a very standard linear model. Is there any way to go, let's say, to something more complicated?
So, yeah. So basically, yeah, we assume that these variants are independent, and we are not assuming it. We are actually showing it that they're independent. So if you do a model with all the different SNPs,
but also the risks are independent. I was wondering more about the risk analysis.
You mean the risks are not I mean, the individual SNPs are not independent between them. But then when you get to the polygenic risk score, you only choose one from each region. That's where the lasso also comes in. So because
I mean, if you see the question, can you please go?
Yeah, sure. So let me just so that formula. So there's lots of different ways in actually calculating the polygenic risk score. This is the most simplistic actually. So you mean this one here, right? No. So this one here, indeed is quite simplistic, but so the SNPs that you put in the formula, we tested them that they're independent from each other. So that's why they're additive. The effect is additive. So we don't go into 2 or 3 way interactions. Other collaborators have actually worked on interaction analysis between the SNPs, like 2 or 3 way interactions. But then you need, it gets so complex, and you need larger sample sizes. So there's not any known robust interaction associations as I know.
At least there is a way to control.
Yeah. So here, is the way also with lasso is basically a dimension reduction, and basically, you know, for between each SNP, you know the correlation. So you try and get SNPs that are uncorrelated into the model.
So you choose, let's say,
Yeah, you choose one. So if you go, so for example, here, so this is one region, for example, from, sorry,
What is your mouse?
I don't know, it doesn't come up.
Yeah, we can't see the slide that you're talking about now. Don't know. It
Can
seems
you
to
see
have
this
got-
now?
Yes, now we
Okay,
can. Thank you.
So here, for example, from this region, you will select one variant into the model. And this is based on the correlation structure and other factors are coming in. And from here, you will also select only one. So you try to reduce the problem. And if you put variants that are correlated, you actually do a conditional model in order to put them together to get the conditional effect size in order to be used. So you don't double count, basically. Yeah. But there is a lot of, there's a lot of studies that are trying to do the gene and gene interaction. So basically SNP interaction. But then if you have 11 million variants and you do the interactions and then you go to 2- or 3-way interactions, then it's, yeah. But if you have an idea on how to do that, then we are more than happy to.
Can I ask how do you combine the risk from germline data to the risk of, say, smoking?
So that's I should have had one slide on that, but there's different models that are actually being used. I mean, I don't have one here, but so here, so this what we did was using logistic regression again. You put the odds ratios from the different risk factors. So each risk factor has some established sort of, if you see here, I mean, so this is smoking, PRS, PARUS, menopausal, height, family history, breastfeeding, and so forth. So you have, so each of those factors have their own risk estimates that were calculated from previous studies. And you can actually put them together in a model that, and you can actually get the estimated risk for each individual based on those. But there are other, of course, there are more complex methods as well to do it, and some AI algorithms as well. But for this purpose, it was more using logistic regression, actually.
You related this risk, this breast cancer risk to other diseases?
Sorry?
For example, or is there any relation between other diseases?
There are some genetic correlation. I mean, so it depends on the relation. I mean, there's sort of some studies called Mendelian randomization studies, where actually you use this genetic component for one disease and try and see if you can predict a different disease. So there is some, I mean, BMI is sort of associated with breast cancer. So there's also some that are causally related, and there you can also do some genetic correlations between different traits. So put different cancers together and see how much of the commonality between the different diseases as well. Yeah.
Do these results for women also translate to male?
To be honest, there is, sorry, I will say in plural, but in the consortium, there are some studies on male breast cancer. Yeah, there is a small, like 1% of breast cancers is quite rare, and quite a lot of it is due to pathogenic variants in the high-risk genes. But there are also, so the new study, the Confluence study, they have breast cancer in males, and there's not very, I mean, there's not much known because it's so rare. So they're going to explore how this works.
That is the same genes.
The high risk genes, yes, but for the SNPs is yet to be known because there's not that many. I mean, it's a small subset of these. And if you imagine that for like female breast cancer, we have 200,000 samples, and for male breast cancer, we have 1,000. So it's
so you need a lot of data program.
Yes, yeah.
What about other types of cancers where you might get
There are also other consortia that are working on ovarian, prostate cancer, the different groups, and they do very similar things. And they, I mean, especially prostate cancer, it has a big, the polygenic risk score is quite
Are there
big.
same SNPs that are responsible for those?
Some SNPs they are, but not all of them. But there are some shared variants.
It's not known why.
Sorry?
It's not known why it's the case.
Yeah, no.
From the molecular, let's say, from the.
So there's functional studies trying to see how, I mean, so those multi sort of is really complex. I mean, and everyone that works on different aspects of these things is really complex. So, but yeah, there's that. I mean, the good thing is that if there's like a big interdisciplinary team, and you try to put different expertise together, then you might get to some answers. Okay.
Maybe from the.
Any more questions from the online audience?
Yeah.
In which case, we shall thank Kyriaki for presenting this brilliant research today. And then we'll have some refreshments for those of us who are here. Okay, thank you all for attending. Have a good afternoon.
Thank you.
Thank you.
Sure.